Good day, everyone, especially to you, Mom. So, my topic for today is about Entomopterales and class Hypomycetes, an overview of fungal pathogens. So, introduction. Entomopterales is an order of fungi within the phylum Entomopterymycota, renowned for its role as insect pathogens, with the name meaning insect destroyers. Historically classified under Zygomycetes, they were reclassified in 2007 due to advances in polygenetic analysis. So, characterized by coenocytic hypea and the production of forcibly discharged conidia. These fungi thrive in organic, rich environments, often acting as obligate pathogens that specifically target insects. So their ecological significance extend beyond insect population dynamics. Many species within Entomopterales are being explored for their potential in biological pest control. So offering a sustainable alternative to chemical pesticides in agriculture. So this unique group exemplifies the complex interaction between pathogens and their host, highlighting their importance in both natural ecosystem and agricultural practices. So, life cycle of Entomopterales. So, the life cycle of Entomopterales fungi, particularly Entomoptera, involves several stages that highlight their parasitic relationship with insects. It begins with infection, where a conidium spore lands on an insect and germinates, producing hyphae that invade the host. This is followed by incubation, during which the fungus spread internally and absorbs nutrients. In the sporulation phase, the fungus reproduces within the host, causing tissue damage. Finally, during mummification, the host dies often after being manipulated to climb to an elevated position for spore dispersal. The fungal mycelium then releases spores into the environment, completing the cycle. So, the importance of Entomopterales. Ecological rule, biological control agent, high, high infection rates, sustainable agriculture, research and development, potential for integrated pest management or IPM. So, this is the introduction to class Hypomycetes. So, Hypomycetes, often referred to as conidial molds, represent a class of fungi characterized by their asexual reproduction through the formation of conidia on hyphae or specialized conidiophores. Historically grouped under the terms fungi infecti and Firpacti or Deuteromycetes. So most hypomycetes are now classified within the phylum Ascomycota based on polygenetic analysis. So this diverse group is notable for lacking closed fruiting bodies, which distinguishes them from other fungal classes. So hypomycetes can be found in various environments, including soil, decaying organic matter, and aquatic habitats, where they play essential roles in nutrient cycling and decomposition. So, identification of these fungi typically relies on microscopic examination of conidial morphology, such as shape, size, and color, as the arrangement of conidia. Additionally, some hypomycetes are entomo- entomopathogenic, infecting and killing insects, and are particularly abundant in tropical regions. Their ecological significance and potential applications in biological control highlight their importance in both natural ecosystems and agricultural practices. So, morphological characteristic of hypomycetes. So first is mycelium, second is conidiospores, third is conidiogenous cells, and lastly conidia. So identification of techniques of for hypomycetes. So identifying hypomycetes as class of fungi known for their asexual reproduction through conidia involves a combination of morphological and molecular techniques. Morphologically, the examination of conodial characteristics is crucial, focusing on features such as septation, shape, size, color, and wall texture. The arrangement of conidia on conidiogenous cells and the type of these cells, whether specialized or non-specialized, also provide valuable identification clues. So culture characteristic, while less reliable, can further aid in identification by revealing growth patterns and colony morphology on media like potato dextrose agar or cornmeal agar. On this molecular side, DNA barcoding offers a precise method for distinguishing species. So, ecological role of hypomycetes. Aquatic hypomycetes play a vital ecological role in freshwater ecosystems, primarily as they decomposers of organic matter. These fungi are essential in breaking down allogenous plant litter, particularly leaf litter from riparian vegetation, which serves as a significant source of carbon and energy in aquatic environments. So, by producing extracellular enzymes that degrade complex molecules such as cellulose, hemicellulose, and lignin, aquatic hypomycetes facilitate nutrient cycling and enhance the nutritional value of decomposing matter for higher trophic levels, including detritivores and other microorganisms. So, the conclusion of Entomopterales and class Hypomycetes. So, the conclusion regarding the order Entomopterales and the class Hypomycetes highlights their significant ecological and biological rules. Hypomycetes is primarily recognized for their asexual reproduction through conidia, exhibit immense diversity with over 13,800 species identified worldwide. They are crucial decomposers in various ecosystems, particularly in freshwater habitats where they break down organic matter such as leaf litter. This decomposers process not only recycles nutrients but also enhances the food web by providing sustenance for detrivores and other microorganisms. So while Entomopterales, often linked to insect pathogens, contribute to biological control in ecosystems by regulating insect populations. Their life cycles typically involve complex interactions with their host, leading to significant ecological impacts. Both groups demonstrate remarkable adaptability and specialization to their environments, which is reflected in their very varied morphological forms and reproductive strategies. So overall, the study of these fungi is essential for understanding ecosystems dynamics, nutrient cycling, and potential applications in agricultural and pest management. So that's all. Thank you.